I change this thing, okay, and I click save, I get a message. It's going to restart my WSL. That's fine. We don't have anything running at the moment, and you can restart the WSL, okay? Then it goes and restarts the WSL and all of those things, okay? Yeah, for sure. Okay, you've got 32, so you've got plenty of RAM. You have 32 maximum, so you've got, how much RAM you got? Eh? How is that? How is it 0 RAM? Can you make this one? 2 gigs of RAM, then 16 is enough for what we're doing today.
So that's the default is 50%.
50%, yeah.
So I'm just adding this to the docs.
Yeah, thank you. Depends on flow, but I thought the biggest multi was anyway. But and of course, remember you're always taking. Is it? And remember you're always using one for the negative control, yes. And if you've got the positive control material, then one for the positive control. You know, we don't always have the positive control. We don't always have a positive control like material. You know what I'm saying? Because we might not have that. But if you've got a good sample, you can use it again as your positive control. You know, in the future. Okay. But yeah, okay, great. So. Here's my in the data, let me see if I can make this bigger. Can you make the view? Is there a way to make this bigger? Zoom in. How can you zoom in on the Explorer? Maybe I can try.
Screenshot? Zooming in the screenshot?
That's possible. I think so. What? They hold down control. Control and control. Oh, there you go. There you go. What's it doing? There you go. So all of these things were on the stick. Okay, I want to highlight this folder. I'm going to make this big. I'm going to say extra large icons. No. Zoom. Zoom, 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 zoom. Ah. This one. It's called MPXV normalized. Okay. When you unpack the data, everybody should have a folder on their computer called MPXV normalized. Okay. It is where you unpack the data. So in documents or wherever you happen to unpack the data. Okay, can you look for this folder, please? I'm just going to show. Inside of this folder, it's got files named like this, SRR something or other. Okay, but just look for MPXV normalized. If you found it, put up a green sticky. If you found your MPXV normalized, put a green sticky. Okay. This, I think we are all green in terms of finding the data. Look, in terms of running the Epi2Me, yeah. So when we're running the Epi2Me, we'll talk to you guys specifically because it's looking slightly differently on your computers. We've all found this data, right? Okay. This is Mpox clade 2 data. Okay. I'm using clade 2 for the training because it was not easy to get clade 1 data. Okay. But you guys will be generating so much. Yeah. Okay. Well, we hope not, but you probably will. Okay. In fact, if you're in Cameroon, you're probably going to be generating both clades, right? You guys are special like that. Cameroon. Okay. First thing, And my screen is a bit small. I'm going to try and make this bigger.
Other way.
View list. View details. Okay, maybe not all read this. I've normalized these. I took bigger files. These were sequenced on a nextclade. I made them smaller for so that we can analyze them easier, okay? That's why it's called normalized, okay? And so the biggest files are here around 13, why is it going dark? 13 megabytes. 13 megabytes here, 13, 12, 2, 5, etc., right? And immediately when we've got this is amplicon sequencing of Mpox virus, right? Which means it's amplifying the virus only. Looking at the screen, which samples do you think are bad samples? Which look like bad samples? Just look at what you can see on the screen. I know you might have to peer closely, but just look here. What do you think is a bad sample?
That's the small size.
The small one, right? Now, I took these from the paper where they first described the Yale Mpox primer schema, okay? Those ones that are very small were taken from very high CT value, 36 CT or something like that, where there's almost nothing to, yeah, okay? So we can immediately see quality control, keep an eye on the file size, you know? If it's very small, something might be wrong, okay? Then I want us to choose, the other thing is, of course, this is Illumina data, so everything is paired. 1, 2, 1, 2, 1, 2, 1, 2, okay? For our first run, I want us to choose one sample, okay? So I'm going to go into my computer, I'm going to go into my, I don't think I'll be sitting, I'm going to go into my documents, I'm going to delete this, and I'm going to make a new folder. I'm going to call it Mpox_1. So now I have a new folder called Mpox1. The name isn't really important, but the thing is, I only want to put one sample for the first time I run things, just to make. Okay, so now I'm going to go back to here, and I'm going to choose I'm going to choose one of the samples that has a nice size, okay? So I'm going to choose maybe this one, the 1 and the 2, okay? Just 1 and the 2 from the same thing, and I'm going to copy, and I'm going to put in my Mpox1. So can everybody make a folder and put one sample in that folder? It can be in your documents or wherever. Just call it Mpox, call it something like Mpox1. Doesn't matter where it is. Just so long as it's only got one sample. Okay? And when you've done that, so take down the stickies. And when you've done that, put up your green sticky. Take down your stickies. Are you already done? Going at the moment. Central Africa is center of the country is representing. Yeah. I know you guys want Central Africa, but you can't say. You're like almost down in the door. To run the workflow, but I need to put this on the screen. I'm going to put it in how to share this with people. Kirsty, can you paste it to the participants' WhatsApp group? That's going to make life easier, okay?
What are you pasting?
What we're pasting is this. Just getting my screen ready. Well, I put this also in a Word document, make it big. Talk to me. Why are we pasting that? No, we must not paste that. Huh? Sorry, I typed the wrong thing here. Undo. Reload. We go to my Epi2Me.
Send you the link to test. Send you the techniques. Send you the link.
Me too.
I'm just going to delete my workflow and bring it back. Hang on, just there's Epi2Me there. So I just need to do something on my side so that I can, in the same situation, resolve, select, delete. Oh, I'm just going to delete the things that I've currently got. I'm just going to delete this one so that I'm in the same position as other people. How do I delete this? I can't delete it. Oh, okay.
Oh, they already have this link.
They do? Okay.
Yeah. Sorry. But I've sent it to them via WhatsApp.
Okay. So that link over there, okay. If you're using Epi2Me, when you start with Epi2Me, the first screen you see is this screen. Not you guys with the 5.1, but everybody else. You see this screen, okay? Then the second one, where you say launch, is where you got a list of workflows, okay? And now Epi2Me is made by the Oxford Nanopore company, and there's a whole lot of built-in workflows that come from Oxford Nanopore, okay? Things like 16S and so on. Is everybody following me and seeing this screen? For you guys, it will look a little bit different, okay? Are you seeing this screen? Yes? This side? Okay, cool. Now, the thing that you want to do with the link, you want to import the workflow, okay? So I'm going to go back to my document, and I'm going to copy this. And I'm going to put the whole thing in there. Okay, now apparently that's been sent to you on WhatsApp, and it's in the document that has been shared with you. It's a long link, but make sure to get it here. https://github.com, Arctic network, artic-mpxv-illumina, okay, NF, okay? I'm going to put back to the MS Word so that you can see it bigger. This link, when you go to import workflow, this is the link that you want to import. Link in here, or you type the link in here. Okay? Now, you guys, your one already set up. Yours is going to look slightly different. Let's just see how it looks. So you go to Epi2Me.
In this doc,
Can you open
I made a comment.
Okay, so this is to run the workflow. Okay? So you can just go to the workflow that you've downloaded. It is here on your list. Then click it. You see? And then you get this page. Okay? This page that describes the workflow, right? You are seeing this, you are seeing this. Okay. Then you click the button which says launch. Okay? Then you see this. After clicking launch, you see this, right? I want to go slowly so that everybody keeps up with me. Okay? Okay. Is it going? Going? Yes? full of samples, and it will process each sample in the directory. Okay? Samples is two files, the forward and the reverse, but it will just process a whole directory. That's why I had you make a directory with just one sample, because otherwise I don't want to process all of the samples, right? So I'm going to click here and go find my folder, documents. I'm finding my folder with the one sample in, okay? Okay, so you're just finding that for that one. The input folder with the one sample. Select folder, and then it shows you here, it goes green, and it's got something selected, okay? Then you want the Mpox 1, the Mpox 1 with just the one sample, okay? Just give me a few minutes. And so go back one level up. You must purchase that part. The folder. Not the Mpox, where's the Mpox 1 folder? There. And now just click selection on your set. Is the primer scheme, okay? Now yesterday I mentioned primer scheme name is always the organism, and then the name of the primer scheme is version, and then which reference you are using, okay? Because the primers that you get and you use in your sequencing reaction, the thing that you put in your the actual primers that you put in your plate where you do the PCR, that is MPXV Yale 1.0, okay? But when we're doing the analysis, we can be doing the analysis with regards to one reference or another reference, okay? Clade 1, clade 2, clade 2B, etc., okay? The default here, I'm actually going to talk to the author of this thing because I don't think there should be a default. I think that it should be read until you actually choose something, because you don't want to accidentally choose the wrong scheme, okay? So now, what are we dealing with? What did I say? This is a clade 2 sample, okay? There isn't yet primers. There will be primers in the future, but there isn't yet primers for both clades. That works. That is, there's not yet systems designed for working on both, okay? Because they're too different to each other, okay? So we choose here, and we look at, there's all of the different primer scheme names, okay? Yale, Riggs Hospitalet, Arctic, BCCDC, whatever, okay? So I know you guys have been, even though you probably don't have that name, but you've been working with the Erasmus primers. The official name is now Riggs Hospitalet for Erasmus, okay? And in the IMAP kits that African CDC will be giving you, it has the Yale primers, the Yale version 1s, okay? There is a Yale version 1.0, where there's a new version of Yale being developed, but it's not in the kits yet. It's coming. Yeah. I've added some extra primers, yeah. And there's actually a multi-clade primer scheme in development from the Arctic people. So we choose the clade 2. So we're choosing the Yale 1.0 with the clade 2 reference, okay, as our primer scheme. The other stuff is if you've got a different primer scheme that's not listed here, but we don't. Okay. Now, behind the scenes, what this is selecting is two files. Okay. The FASTA reference, the genome that we're going to compare against, and the bed file that describes the primers and the positions. If you remember yesterday, I talked about the bed file, which describes which reference and at which position the primers are found. Okay. So primer scheme selection must be clade 2. Then the next one is, where do you put your results? Okay. Now, if you do not select the output location, it will put the output somewhere inside of the Epi2Me labs directory, and it will be quite difficult for you to find afterwards, because it's in like some funny long name of where it is. Okay, so I always choose an output location. Okay, so I click here, and now I need to make an empty folder. Okay, so I'm busy choosing the output location, but it doesn't exist yet. I need to make it still. Okay, so I make the new folder. And then I select the folder, okay? Can everybody double check that they've made a new folder for the output location, and they did select the folder? Okay? Is your thing working again? How? Oh, it opens into the background. Let's see. Okay, now for you guys, there's an extra option for the output, but I'm going to show you in a minute. Okay? So once you've got your output location, okay, everything good? Everything good? No? What's not good? Tout c'est bon? Too good. How can it be too good? You're going to smile so much your face will split open like a watermelon. Huh? It's really beaucoup. Okay. Okay, the next option that you need, just checking that you've got the SOP put up. Are you ready? No, I want to go slow because you must get. Okay. Governor is coming right there. You just creating the photo.
Can you scroll up for a second?
This is at the top.
Oh, is it for the primary scheme section? Is the top maybe? Oh, slowly.
Yeah, so you can actually, so in theory, we could actually use the same analysis pipeline for other viruses that use Amplicon sequencing. It's not supported yet.
So if that workflow works for
Marburg, for SARS-CoV-2, for whatever.
oh, okay. So you can
Anything that's Amplicon sequencing. And has a single segment. Life gets complicated with your multi-segment viruses.
and then you said you had monkeypox 1 and 2, so you essentially use the same workflow.
But just with different references, yeah. Because the inferred ancestor of Mpox 1 and 2 is very far back in time. So Mpox is first seen in humans in 1970, right?
Yeah.
But the split between clade 1 and clade 2 is probably like more than 100 years ago. So there's quite some substantial difference between the two things.
So they would eventually develop two separate workflows for clade 1 and 2.
No, because the bioinformatics technique is the same.
Okay.
It's just that the data input is different, like, you know. Yeah, because you know in bioinformatics, so much is actually general. That, like, if you are assembling one plant to another plant, it's still the same plant assembly process. But it's just that because of the so when you look at metagenomic ones that are not Amplicon based, you can use the same workflow for just about anything. So if you've got, you know, so yeah. I hope that it gets even more so over time, because I would like to be able to teach people basically 1, 2, 3, 4, 5 techniques, and then have them have a very broad base of techniques that they can apply for different things. You know, because like, yeah, anyway. Okay. Push on, step. Push that down. Okay. You need a name for your output file prefix. Can be anything. I'm just going to call mine 1. Okay. So just put anything in that field there. On your side, I want to see what your output screen looks like. The last thing we need to choose is for the squirrel. Which kind of a squirrel do you like? Do you like a red squirrel, a gray squirrel? Do you like this little albino white one? No, I'm joking. What you mean for squirrel? Squirrel is also needing to know which clade are you analyzing. Okay? So for squirrel, you must select an option, and we must select clade 2. If you don't know what clade it is, at the moment you have problems. So, yeah. This is a damn good question for my colleague over here. What do you do if you don't know what clade you've got? The problem, yeah. But remember, we're now moving towards a world where the trinders, Yale was never designed for clade 1. Neither was Erasmus, was designed for clade 2. All of the trinders were designed for clade 2. Okay? So, we're moving towards a world where we have universal trinders that work for both clade 1 and clade 2. Then what do we do with the analysis? I think what we're going to have to do is do the analysis and see what it looks like. Because if you take a clade 2 and you compare it to a clade 1 reference, how many mutations do you expect to see compared to if you take a clade 1 and you compare to a clade 1 reference? With the clade 2, you're going to see so many mutations because it's very different to a clade 1. So because you can always redo your bioinformatics, it doesn't cost you anything. Okay? So, so then you have to, that's why I always say, look at the number of mutations. Okay? Remember what I said yesterday? Was it yesterday? It's already, time is flying. How many mutations per, naturally occurring mutations per year in Mpox virus? 2. Okay? How many per month in 2 mutations per month in, yeah, no, you went here, but 2 mutations per month in SARS-CoV-2, 2 mutations per year in Mpox virus when it's in its natural host, whatever the hell that is. Somebody please find out for me. Okay? In human-to-human translation, human-to-human transmission 6, about 6. If you have sustained human-to-human transmission. Now we're not having, this is new, I'm saying 6 for now, but this is a new world for us. We don't know what's going to be looking like. If we carry on, Mpox never used to have such long, Mpox, they used to say that the transmission chain is 7 people and then it ends, maximum. But now we're seeing these long human-to-human chains. And we don't know what the future holds. You know? Okay. I think we are ready. Are we ready? We've got everything filled in. Everything is green. Click Launch Workflow. And then it opens up this window and you must click Launch a second time. Launch. Launch. And now it does some things. Brum, 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 brum. And then you see this screen. Okay? If you don't see the screen, put up a red sticky. If you see the screen, put up a green sticky. If you're still busy, take down your sticky. Is it launching? Have you already launched this a long time ago? Probably. Huh? We know these people who don't have the C, they don't have any chill. There's no chill there. Yeah. You must say yes. Say yes. What do I say? He doesn't need it, but okay. We agree. Okay. I don't see any red. I'm going to do it down. Okay. Now what we're seeing here are the different steps of the workflow. Okay. The first step is indexing the reference. This is a technical thing. Okay. Reference, the reference sequence that you're comparing against. Which format is the reference sequence in? Which file format is the reference sequence? Look, bed is a primus. FASTA. Okay? FASTA, yeah? The genome reference is in FASTA format. Okay? It makes an index so that it's quicker to search through the thing. So that's the first step that it does there. The second step that it says is fetch host. Okay? Fetch hostile something or other. Okay? Everybody should see it go past that quickly. Okay? Has anybody's workflow stuck on the second step? The fetch hostile reference. Anybody have a workflow stuck here? It's stuck there. Then there's something wrong with your configuration. Let me see what you've got. No, it's faster. It's green. What's stuck? Oh, sorry. Oh, you put it, that's it. Fetch hostile is green. That's green. Yours one. Green is really good fuzzy. Okay. It's kind of hard to read the screen because the names are long. Okay. This tool, which is called hostile, okay, is hostile. It doesn't just mean that this tool wants to fight with you. Okay. It's called hostile because it's for removing the host. Okay? But if you need to compare, if you need to remove any human reads, right, then you need to have a copy of the human genome to compare against. And that's why we preloaded it onto our computers. That's why we did this stuff this morning of preloading hostile. So if you go home and you've got a different computer, you must, if you want to save bandwidth, preload the hostile data onto your computer. Yeah. Stop with error. That's an interesting. Where is it gone? Logs. If you look at the logs tag, you actually see the detailed log file of everything that's happening. Okay, so go switch to your logs tag. And you can see it starts off with the parameters that get set, and then does some other stuff, and it says here after the parameters, it says submitted process, okay? And here we can see in detail which things is it doing. Submitted process, prepare reference, fetch hostile reference, etc. I want to say now, yours there must be an error. Okay, yeah, up here. Unable to find anything. Okay, there's a problem there. Load view. Then this is the error. So you see here, the file exit is unable to find this image. Load view. Okay. Pulling from hostile, died to the download. Okay. Then what's the error here? Okay, I've got it. I think I know what the statement of the problem is. I don't know how to fix it, but I do know what the problem is. The space. Your username is spacing. Your username was to spaces. A lot of these tools are not Google Spaces names. So, what can we do to fix it? Your username. We need to fix the workflow to deal with it better, but for now, we're making new user on the. but can we make new use of it? Directory are useful to help people debug the problem. Okay? Then each step has its different output files. Okay? And but I just want to highlight two output files immediately. Okay? The first one is called run1.qc. Okay? 11.qc is a CSV, comma separated value file. Okay? Now, I have LibreOffice installed on my computer, so that is what opens comma separated value files for me. For people who have Excel, I don't know, I'm not an Excel expert. Can you open in Excel the CSV file?
Yes.
Okay.
Which one do you live in?
No, Excel used to have problems with this. You used to have to go data import and other weird stuff. Yeah. Okay, so if we look at this file, it just gives us some basic statistics. Just bring the screen to the right place with my mouse going. Basic statistics about this analysis, okay? And this is kind of boring because we've got one sample, right? But if you've got many samples, it's going to be more interesting to see all of the samples, and particularly we can see which percentage of bases were covered. When they say covered, they mean with at least 10 reads were matched onto that position in the reference, okay? So we see here that 93% were covered, okay? And 6% were not covered. They're basically matching. The number of reads that align, 296,000. And then the longest one of ends, okay? The longest no end one, the longest stretch where there's no ends, okay? And this is 126,900 and something. Remember, the Mpox genome is about 200,000 letters long, okay? That means that there were still some ends in the genome, even though we've got a good quality sample, okay? But remember what I said about the Mpox virus. The Mpox virus is more complicated than SARS-CoV-2, and there's some parts of the genome which are difficult to analyze, okay? Okay, so that's just some basic quality controls. Now for the next thing, who here knows what nextclade is? Nextclade? If you've worked with COVID-19, SARS-CoV-2, you might have used nextclade. Okay? Nextclade is from the same group that makes nextstrain. Nextclade is Nextclade. Nextclade. Okay? It's a website. So if I go to my web browser, and I look for nextclade, then here's the nextclade website, clades on nextclade.org, and I can take the output file from my Mpox1 output, the all consensus file, okay? You see, it says run1.allconsensus because I said that the prefix for my file should be run1. If you chose a different prefix name, your files would be called something else. Okay? So if you make it called your prefix 1, then it will be called 1, etc. Okay? And if I load this run1 all consensus is a FASTA file of the consensus for this one sample. Okay? Before I show it to you on nextclade, remember the approach that we take is we take the reads we map them against the reference, and whenever we find variation, then we insert that into a copy of the reference. Okay? Where we find, but if only if the variation agrees all with each other. So if we see a variant at some percentage, normally it's something like 75%, then we'll insert into the reference sequence, right? If we don't see enough reads, what do we put into the reference sequence? An N, okay? Because that means we don't know what's there, okay? And so that means that the size of the consensus is approximately the same size as the initial reference that we used. We've just inserted the differences that we got from the sequencing data, okay? So it's a FASTA file, and if you run more, if you had more than one sample in your directory, if you analyze multiple samples, then this all consensus file will contain one entry, one consensus entry for every sample that you analyze, obviously in FASTA format, okay? So this thing over here is maybe one of the most important files that you get out of your analysis, because now you've done your sequencing, you've got your analysis, you can then do lots of things with it. You can then, this is the file that you're going to analyze to figure out what clade we've got. Is it a clade 1A, a clade 1B? Is it 2, is it B.1? What is it? Okay? All of the other files that are generated, I consider them basically for quality control. But this is the one that is for all the downstream analysis. Okay? So I've selected the file. My screen is not scrolling. It should be showing you at the bottom. Why is it not showing?
Smaller?
No, I'm going to try and make my screen smaller. Okay. Normally it's showing here at the bottom. I don't know why my screen is not scrolling down. Ah, there. Good. So you see it's now been, when I selected file and I added it here, you can, with nextclade, you can analyze more than one file at once. Okay? You can load multiple files into nextclade at once. And by the way, the nextclade analysis runs in your browser. So the data that you've got never leaves your computer when you're using nextclade. If you're worried that somebody could be stealing your data, it never actually leaves your computer. Okay? And then when you, on this side, it's which reference data set should I use, okay? nextclade has a nice feature these days. It will try and guess which is the correct reference to use in your analysis. It will look for which reference is similar to your consensus sequence, okay? So if you've got a clade 1, it will recommend a clade 1, etc. Yes? What? Yeah. Because this one, you see what, actually I want us to start from 0, right? If I reload this page with nothing, then I'm just going to, choose a different reference. This is a starting point, okay? I'm going to choose SARS-CoV-2, okay? So now I'm going to switch this button, suggest automatically on, okay? Now I'm going to select my data. And now it says here, oh, this is wrong. That won't work. Change reference data set. Okay, so now I'm going to go.
There was a resuggest option.
The resuggest. Okay, let me go back to that. Okay, start again. I'm going to just again make a change reference data set. I'm going to make this SARS-CoV-2. I'm going to switch this to that. And then I'm going to say there, resuggest. If I click resuggest, it chooses one. And by the way, this is Mpox lineage B.1, okay? Just to illustrate again what we're dealing with. Where's my Mpox? I'm just looking at all of the Mpox, okay? Just remembering the terminology, right? Because by now we all know what a delta is and a beta and a omicron and all of those kind of things, okay? This is what the diversity of Mpox looks like. Clade 2, clade 1, and B.1 is down here, okay? This is colored by the clades, but if I color this thing by the countries, if I look at the map, okay, B.1 is this red color over here. We see that B.1 is the one that's found almost all over the world. So the outbreak that we have in the world is mostly a B.1. I mean, outside of some good endemic countries, is mostly a B.1 outbreak. Okay, it's a subclade of clade 2. But then and you'll notice that here, there's B.1, there's A, there's all of these kind of things. In clade 2, all of these subclades have names. In clade 1, we've only named 1A and 1B, and we've not got names for the more detailed divisions. Maybe over time, we will say that there's a clade 1C or something like that. We don't have names for it yet, okay? So it chose, based on our sequence, it chose B.1 as the reference. So let's go do some analysis. Run the analysis.
A question, that auto-suggestion, is it fairly accurate?
Yes.
So, on average,
what the auto-suggestion is doing is it's taking all of the references and it's lining up your sequences against the against the references, and it's asking which is the closest. So if you've got a sample, so let's think about when would that not be accurate? When would you not get accurate results from the autosuggester? I can think of two scenarios.
It's like mixed samples are contaminated.
If the sample was somehow in between, maybe mixed or something like that, but the other thing is if you've got a poor quality sample and you don't have a lot of information about whether it's close or not, then the autosuggester is not going to work. Like if you only got 10% of your genome, then there might not be enough. Okay. So yeah. Public health importance. You're the public health person. You're the one who's doing it. Yeah. So that's a damn good question. Why are we even bothering to sequence Mpox? Cyrus, why? So in terms of James was going to give a talk on genomic epidemiology next Monday, but I say that there's a few reasons that we in general want to sequence things, okay? The one that we want to know is how many different transmission chains do we have in the country, okay? What? So we can look at the mutations, but it's not, remember, the mutation rate of Mpox virus is slow, so it's quite difficult. As I was saying, if James had Mpox, no, but we're going to do it again. If James had Mpox and he transferred to Eve and to you and all of the way around, and it's not infects me, my bet would be that the virus would be identical, because this is not an RNA virus. DNA virus mutates slowly. So yes, kind of. I mean, and when we say transmission chains, we're probably looking at quite a big scale thing. Like we know that 1B and 1A, they're quite different, okay? But the second reason is sometimes there's an association between variant and disease severity, okay? So we saw that with Delta, that Delta seems to be more severe for COVID than Omicron, okay? And we're actually seeing that the evidence is still being accumulated. but there seems to be evidence that 1B is less severe than 1A. And that seems to be related to the loss of a gene. One of the things that's different about Mpox virus compared to COVID is that entire genes come and go. It's like this picture here. Every little block over here is one gene. So you see how many genes Mpox virus has compared to COVID, which has got like what, less than 20. I can't remember how. If you add them all of the things together, you've got off 8, and then you've got the polyprotein being cut up. I can't remember how many proteins there are in SARS-CoV-2, but it's not many.
No, around 10. They're actually very few.
10, yeah. Compared to, look at all those proteins there. That's why I said, to me, Mpox is almost like a virus that is almost like a bacterium. It's got so many different genes. Okay. Yeah. So these things are being, and these are being added and changed. And so there's a relationship between severity. And then the third thing is, but I don't think this is going to be so important for Mpox. I don't know. The question we always ask for genomic epidemiology is, does this change make something more transmissible? So if you remember with COVID, the D614G change in the middle of 2020, we think made it more transmissible. But then definitely alpha and beta led to more transmissible forms of virus. So we don't know if any of these mutations in Mpox are going to make it more transmissible. So that's another one of the key things that we look for in terms of, so we always do a correlation analysis, because that's why with genomic epidemiology, we cannot do it with only genomes, right? We're always collecting clinical data and virus data and comparing the two and trying to find trends. And the big problem that we have in our continent is that we're not collecting enough of either kind of data. In most of our countries, we have poor clinical data collection mechanisms because many people are going to health facilities that are not actually having the maybe electronic health records or data collection mechanisms, right? And we have poor genomic. We are missing most cases. You know, we have poor diagnostics, but we try. Okay, we try and do the genomic epidemiology. Okay. So what do we see here? Okay. Let's look at the screen. Okay. This side is the quality control metrics. If you've used nextclade before, you've seen this before. Okay. And what this means, let's go through them one by one. N. How much of the data is missing? Okay. Now, we would love to get 100% of the genome. Okay. But we are missing, we have got, according to the quality control, 93% of the genome. Okay. To me, 93% of a 200 kilobase genome, that's quite a lot. Okay? That's quite a lot. But it's yellow to say they wanted, their limit was, they say that it's bad if you're missing 10% of the genome. Okay, or a little bit over 10% of the genome. So they've got it yellow, not red. But in my experience, lots of Mpox samples, we don't get a complete genome. Next problem. You guys are all being given the Yale version 1 primers that are designed for Mpox clade 1. They're already not great for clade 2. Yale version 1 is already not great for clade 1. Okay? They miss some parts of the genome. Just like we had to change primers during COVID. But the problem is if you wait for the new primers, you're not going to be sequencing this year. You'll probably wait till next year sometime. Okay? Takes time to get new resources. The next thing, the M on my screen. M is for mixed sites. Okay? It's mixed. Okay? Now, yesterday we said two things can cause a mixed site. Let me just go back to my slide. If I can find that slide. Do I have that slide somewhere? Yeah, but finding things on my computer is... maybe. There you go, I think it's this one. Oh, one. Another untitled presentation. I so need a PA to organize my slides. Oh, it is in the drive, right? Yeah, it's in, see if I can find it quickly.
I'm sending it to you on WhatsApp.
Is it this one? There you go, this one. Here is a picture of a alignment of reads against a reference for amplicon sequencing. Here is a not mixed site, because it's all the same variant. Here is a mixed site, okay, which is a mixture of the blue and the yellow variant, okay. We discussed this yesterday, and we said that that can either be the result of contamination, which is why the negative control is so important, or it can be a mixed infection. But if mixed sites are seen, then nextclade gives you a warning if there's too many mixed sites. Because even if it is a genuine infection with two different viruses at the same time, you should go and inspect that. You shouldn't just, normally you should not be seeing mixed sites. Okay? Then we're on to P, private mutations. Okay? So what is a private mutation? Okay? What that means is when we see a new sample, we do not expect to see lots of new mutations. If you've got a new sample of Mpox virus and it has 100 mutations compared to the reference you're looking at, typically that will be a mistake. But this is relative to the reference. This analysis is for B1. B1 is a clade that emerged recently. If we chose A, which if we chose 1, clade 1 as our reference, the clade 1 reference was collected, I think, in 1996. So we expect to see more mutations in a clade 1 because we're comparing to a virus from 1996 than in a clade B1 where we're comparing to a virus in 2022 or something like that. Okay? So, but private mutations, they basically, you shouldn't have too many unusual mutations, okay? Then mutation C for mutation clusters, we don't normally expect to see lots of mutations right next to each other. And if we do see lots of mutations right next to each other, then that often implies bad quality. Because, like, why would the mutations be stacked up like that? Okay, that's what C is. F is for frame shifts, okay? Who here knows what a frame shift is? Most of these things are easy to explain, but frame shift is a little bit more difficult. Who here knows what is a frame shift? Some of you guys are virologists by background. Don't you know what a frame shift is? Okay? When you're translating from nucleotide to amino acid, 3, 3, 3, the codons, right? But what happens if you have an insertion and a deletion that is not a multiple of 3? That's called a frame shift. And normally that we don't expect that, because if we see a frame shift, that means that the gene does not get translated correctly into a protein, right? Because you can imagine that I think I must find a picture of this. But you know, if you basically like have a gap of 2, then then that you can end up with a multiple protein. And if that protein is essential for the virus to survive, you don't expect to see that in the virus. Okay? Problem is, again, Mpox is a little bit more complicated, right? It has so many genes. We don't know if every single one is essential. If we see a frame shift in SARS-CoV-2 data, I would almost certainly believe there's a mistake. Unless we're dealing with, there are some arguments around ORF8 in SARS-CoV-2, but if we saw a frame shift in the S protein, and we've got a malformed, messed up S protein, that's impossible with SARS-CoV-2. It cannot survive with a frame shift, right? Mpox is a little more difficult, but you in general shouldn't see frame shifts. And then stop codons is when a similar thing, but where a mutation happens that causes a protein to be truncated, and a new stop codon gets inserted into that. Again, we're not expecting to see that. And this, compared to this reference, which is a 2022 reference, just to illustrate, just zooming, just scrolling a bit, we have this picture on the here shows the gray, shows where we are missing data, okay? We're missing data at the start of the genome. Remember that the start and the end of the Mpox genome has got inverted terminal repeats. So normally we don't analyze the start and the end of the genome because Illumina short read data is no good with repetitive sequence. So that's not surprising that we've got a missing piece over here. And then we have two positions which are non-ACTGNN. And then over here we have, compared to this B1 clade genome, the sample that I've got was actually collected, I think, last year or the year before. We only have one mutation compared to the reference. This is almost identical to the reference, okay? So we've sequenced almost the entire genome, and for our efforts, we've got one mutation, okay? This is not unusual, as I say, because this is a DNA virus, okay? But nextclade, you should always run your data to nextclade. It tells you what lineage it is, it tells you how many mutations you've got, it tells you, you know, the quality results and all of that kind of stuff. Nextclade should be one of the first things that you do after you've got your sequence results. We have afternoon coffee.
Yes, it's ready. So.
I'm sure it's ready. Okay, questions? Throw them all at me. Come here. Oh yeah. Stanislas, can you explain the importance of the nextclade results in français? Yeah, the quality control. Can you go through these different quality control metrics en français? What is the missing data, the mixed sites, all of those things? You can just give a quick rundown of what the quality control means. If you need any explanation, I can explain to you. Okay.
Missing data.
Yeah.
Missing data. Par rapport au génome de référence, on s'attend à ce que, quand on est passé au séquençage, on ait des sites où toutes les bases sont données. Le missing data représente le nombre de bases pour lesquelles qui n'ont pas été insérées. Par exemple, ce sont des bases inconnues. Donc, dans notre génome consensus, on se retrouve avec 12 316 positions pour lesquelles on ne sait pas qu'est-ce qu'il y a là, comme lettres A, T, C et G. Donc, c'est ce qu'on appelle le mixing data.
Yeah. The M, mixed sites.
The mix site.
Why is it bad to have mixed sites? Because it is a sign of possible contamination. Remember? Because if you've got a mixture in one position, let me just go back to the slide. We should not be seeing this, right? A mixture of two different variants in the same position. So that's why mixed sites is a warning. I'm not saying it's, none of these things are, with quality control you have to always apply your mind. But that's why mixed sites, if there's seeing lots of mixed sites, it's an error. One second.
peut se
warning.
retrouver où on arrive à des positions où on arrive par exemple autant de A. Donc, normalement, lorsqu'on veut construire le génome de référence, on aligne nos 8 sur le génome de référence. Et à une position donnée, on regarde le nombre de A, par exemple, qu'on a eu, si ce sont des A qui sont à cette position, et le nombre de C. S'il y a autant de A que de C, on risque de ne pas pouvoir conclure. Et donc, dans ce cas, ici, par exemple, on dit que le nombre de 1000, c'est bon, ça veut dire qu'on n'a pas retrouvé ce type de position. C'est private mutation.
Okay, private mutations is we expect, so if we look at the tree, okay, if we see compared to the reference, we expect there to not be like too many mutations that nobody in the world has ever seen before. If you know what I mean, because like with SARS-CoV-2, right? We expected everything to be like we saw before, but a little bit mutated, not like 100 mutations different. The same thing with Mpox virus. So private mutations are mutations that nobody in the world has ever seen before. And the thing is, if you see too many of them, it's a warning sign because get it.
Une private mutation, normalement, lorsqu'on prend le génome de référence, par exemple le cas du SARS-CoV-2, vous avez tous utilisé. À une position donnée, on s'attend par exemple à avoir A comme mutation. Ça, c'est ce qu'on espère avoir. Et on se retrouve dans un cas où, à cette position, on retrouve d'autres mutations que le A. Ça veut dire quoi? Par rapport à ce qu'on espérait avoir, on a obtenu autre chose. Donc, ça veut dire que ce sont des mutations qui ne sont pas attendues. C'est cela qu'on appelle des private mutations.
And then can you just explain one more thing about that? Is that this is a B1 reference, which means that the samples are very similar. B1, what is this one? Okay, but B1, let's ignore the terrorists, the keyboard, okay? But the B1 the reference was collected recently. But if you look at clade, yeah, but that's a little bit of a complicated explanation to explain to people, huh?
It still helps.
And yes, I know, but still.
This can be good for translating.
Okay. But I was just saying that like the number of mutations that you expect for clade 1, because the reference was collected back in the 1990s, is going to be more than the mutations you expect for clade B1. Because the
What's yours, Emma?
Okay.
You,
Okay. You
you
want to
can
explain
see?
more?
James.
Yeah. Any other questions?
C'est que pour être échantillon donné, est-ce que les lettres là sont juste à titre indicatif?
Ça, c'est net là.
On sait que par exemple, quand on voit N, c'est qui représente les données non lues. On donne à peu près le pourcentage de données non lues. Maintenant, la couleur, il y a parfois on peut voir le N là peut être vert. Donc vert, est-ce que quand on voit vert, ça veut dire qu'il n'y a pas eu de données manquantes? Parce qu'il faut que je vois la nuance d'abord, les lettres, chaque lettre a une signification. Et maintenant, en fonction des couleurs aussi, voilà, ça c'est par exemple, voilà, contrôle qualité. Donc on sait par exemple N, représente les données manquantes. Maintenant, la couleur peut être verte. Ça veut dire que dans mon échantillon, il n'y a pas eu, dans ma séquence, il n'y a pas eu de données manquantes. Par contre, si nous prenons même P,
C'est du vert.
voilà. Parce que ça, il faut
Il y a une iconographie.
voir. Si on peut voir un peu ce que ça signifie. Donc, chaque lettre a une signification. Maintenant, on va avoir le P étant rouge.
Yeah.
C'est quand ça soit rouge, ça veut dire qu'il y a.
C'est le pas
Yeah.
Voilà.
Yeah.
Mais il faudrait
Pas
qu'on
bon, hein.
comprenne que P représente ceci.
Oui,
Voilà.
et c'est ce qu'il a.
La mutation, par exemple.
Qu'est-ce qu'il a?
Yeah.
Oui, voilà, yes.
Chaque lettre a ça.
Voilà, c'est ça.
Yeah. Okay. So shock letter is one of these things. Anyway, there was a question. And then, yeah. Go ahead. Ella. Yeah. Plus one.
Deux questions, ouais. Two questions. Faut pas le voir.
Stand there.
Oui. Quand on fait de l'alignement sur une séquence de référence, how many mutations do you expect to say that we are in another clade?
In another clade. It's lots. I'll get the numbers by tomorrow. Huh? Adaman, I'll look it up overnight. Actually, what time? Tea time. Okay. Yeah. Yeah, go back to the next slide. Huh? See?
Au niveau de quoi? Filter.
The filter is just to filter if you've got more than one entry. So if you've got like, if you can load 20 different things into nextclade. To filter, like if you go to filter, you can look at, it filters the rows. So we can ask for only the ones, if we look here, but this only, it only makes sense if you've got lots of samples there.
Si tu passes par exemple 200, 300 échantillons, tu vas voir que tu auras un grand tableau, n'est-ce pas? Maintenant, tu peux regarder par exemple le comportement sur un échantillon spécifique. Donc, tu peux utiliser le filtre pour aller à cet échantillon ou bien à un ensemble d'échantillons.
Okay.
Let
Let's
me
get
ask,
time for the tea. Yeah,
let
yeah.
me ask Larissa, was she asking that when you have a mixture of colors, how do you decide, like if you only have green, yellow, if you have red, green, yellow, red, how do you decide which one to pick? I think that's what she was asking. If I'm not wrong.
Let me show you.
So I
Let me
have
show
some
you
bad
something.
data if you need to.
Let me, can we discuss them tomorrow? Okay. Huh? Tomorrow I want to do a couple of things. I want to look at more samples and look at bad samples. I also want to show you what happens if you choose the wrong reference. If I take a clade 2 versus clade 1, what does it look like? I want to show that. We will look at that. We will look at more things. I'm sorry to take more time from the lineups, but tomorrow, up until maybe tea time, we're going to look at more ways to understand your data. Now we only ran one sample. You could have done your analysis for the whole direction full of samples. I just wanted to run one because time was short. Okay? Le café?
Peter, I just wanted to ask you, do you want us to have a quick tea break so that they can come back and do one?
No, I think that we are. I'm seeing people getting tired. Huh? We are not because the